There we go. Hello, everyone, and welcome to the 8th Monthly Orion webinar for 2014. We are honored to have today Dr. Martin Hoffmann-Zapicius from the Fraunhofer SCAI. He holds a PhD in molecular biology and has worked for over 10 years in experimental molecular biology. Since 2002, Dr. Hoffmann-Zapicius has been the head of the Department of Bioinformatics at the Fraunhofer Institute and is also a professor of applied life science informatics at Bonn-Aachen International Center for Information Technology, as well as the initiator and academic coordinator for the IMI project, Taxonomy. Today he will be joined by Alpha Tom Kodamolil, who holds a master's degree in life sciences informatics from the University of Bonn, where she is currently a PhD candidate. She is a researcher at Fraunhofer SCAI, where her work focuses primarily on automatic expansion of computable disease models by developing reasoners. Shweta Bhagawati will be presenting with him. She is also pursuing her doctorate at the University of Bonn and a researcher at Fraunhofer SCAI, where her primary interest is in the field of neurodegeneration. Together they will discuss the topic, Towards a Mechanism-Based Taxonomy of Alzheimer's Disease, Modeling and Mining Strategies for the Identification of Complex Pathophysiology Underlying the Neurodegenerative Diseases. Whew. Please remember that you can type in your questions anytime, and they will be happy to answer them during the presentation or immediately after, probably immediately after if I recall correctly. So I will throw it over to our friends in Germany for this presentation. We'll enjoy the show, folks. Thank you.
Good. I hope you can hear me. Thanks, Lenny. If I could have the first slide, this is Martin speaking. Thanks for the introduction, by the way. And yeah, so my name is Martin Hoffmann-Zapicius. Lenny already introduced me. I'm the head of bioinformatics at the Fraunhofer Institute for Algorithms and Scientific Computing, which has this SCAI abbreviation, which is SCAI. Our computers are, so to speak, a Skynet, but we have no evil plans, be assured. Can I have the next slide, please? The Fraunhofer Society, just as a brief introduction, the Fraunhofer Society is Europe's largest applied research organization. It has now, in the meanwhile, more than 60 institutes. There are 7 institutes actually in the US, 2 in China, 1 in India, and we doing applied research. Typical funding schema of a Fraunhofer Institute is that we have a really little amount of institutional funding and make most of our money with contract research with the industry, in our case, predominantly the pharmaceutical industry, biotechnology industry. And we are also heavily involved in larger public funding initiatives and projects, including that IMI, the Innovative Medicine Initiative, which has already been mentioned by Lenny. Okay, next slide, please. This is just that's something I show always to make people in the US jealous, because we have our own little castle, as you see in this aerial photo. That's the campus we work on, and of course, as usual, in that castle, no scientist works, but only administration and legal. Next slide, please. Good. What is our department focusing on? We have 3 major areas where we do research and development. One is we are quite well established in the field of information extraction in the life sciences, which means we deal with named entity recognition, relationship mining. We do that in a scalable fashion, so it's possible for us to process, for instance, 1 million patents or so. And we do general text and image mining. We use that technology actually quite intensively since the last 4 or 5 years now for integrative biology and disease modeling approaches, and we have a strong focus on neurodegenerative diseases. One of the motivations comes from my personal background. I'm getting older every day, and my risk increases all the time. Start forgetting things, and that's one of the points. The other is really that we all know that Alzheimer and other neurodegenerative diseases, dementia in general, is a huge societal threat to all of us, and I love to work also in directions that address societal challenges. Finally, we have a strong background in grid and cloud computing, high performance computing, and some of our co-workers and some parts of our institute are actually focusing very strongly on high performance computing, cloud computing aspects. So we benefit basically from our ability in-house to scale up information extraction workflows. And the big theme underlying why we are all doing that is we want to make scientific content available for computing, and let me assure you the majority of real knowledge in biology is still in epic, in natural language, in papers, in publications, and not in databases. Next slide, please. So currently the department has about 10 scientists, a couple of software developers, PhD students that I'm very fond of and very proud about. And two of them are with me today and will basically give an overview on work they have been doing. And we have a varying number of master students. I have an affiliation, as Lenny already mentioned. I have an affiliation also with the University of Bonn. And that makes sure that we are in continuous touch with the pure academic world, but we are also working very intensively with the industry, and that makes us somehow a link in the middle between academia and the commercial side of things. Next slide. That's good. We have actually developed a couple of tools, which is one is the most widely used one is called ProMiner. We have won international competitions with that, with sort of critical assessments of the performance of information extraction pipelines, and we are regularly participating in challenges like that. We have developed a interface, a literature mining environment for biologists and medical researchers that makes literature mining easy, and you don't need to have any computer science background to use that machine. And we have also tackled the problem for some while to reconstruct chemical information from chemical structure depictions. So basically, a back translate from an image of a chemical structure into a computable chemical information. And what's very important, and that's something we will talk about today, we are involved in major public research initiatives, and the flagship project currently, which I lead as an academic coordinator, is the Taxonomy project in the context of the Innovative Medicine Initiative, which is a public-private partnership between the pharmaceutical industry at global scale with the European Union administration. Next slide, please. Yeah. Okay. So I already mentioned that we are living between the academia and the commercial industrial side of things, and actually combining scientific excellence with the applicability of our research in the industry is a non-trivial goal. I have been working in pharma industry, I have been working in biotech, and I have also been working in academia. So I know the metrics and the success criteria in both worlds, and that does not always go so well along. It's not totally congruent, and we try really hard to be good at both metrics, basically, for both communities. So in the following slides, we will see then a couple of approaches that we have developed to identify candidate mechanisms for neurodegeneration, and that will be basically the talk of Shweta and Alpha. However, on the next slide, I would like to set the
Martin,
scene.
this is Lenny. I need to interrupt for 1 second.
Yeah.
Could you please go to the left of your screen and click the green screen share icon? I don't think your slides are being seen, because if you're looking at the slideshow, that should share it with everyone. Go ahead and click that.
Oh, I did, but that slide. Those slides are shown by our
They're,
operator, huh?
they're
So
being,
Yes,
yeah, they're being shown by me, Lenny.
but
And
they're not on the broadcast.
oops,
There we go. Perfect. Leave
I
it
could
like
see
that. You'll
them.
be fine.
you can see it now?
I could
Yeah,
see them. Yeah.
you could, but the people who are watching who aren't in the control, who aren't one of us, don't have the option. It's a live feed through YouTube. So we're fine now. So please proceed.
Okay. In that case, you know, you have all my wordy explanations. um, the background for why are we after disease mechanisms, or what does Taxonomy actually want to achieve, is that Taxonomy is all about generating a mechanism-based taxonomy of disease, in particular, in that case, for Alzheimer and Parkinson's disease. That goes back to a paper by Ismail Kolar and John Bell in 2011 in Nature Reviews Drug Discovery, where they wrote a nice comment, basically a statement saying we are classifying diseases in a way that is still following the principles of more than 150 years almost ago, the very early classification for human disease, and we are not including in this taxonomy any of the molecular information that is available, or the mechanistic information. And we're calling for reform of the taxonomy of human disease. And I can only recommend to the audience to have a look into that Nature Reviews Drug Discovery short paper by Ismail Kolar and John Bell. They say we need to basically set up a new taxonomy which allows us to identify patient subgroups which have to be treated in a subgroup specific fashion. Just see and just read that the slides are not visible. Sorry for that. But we could probably upload the slide sets or so with the video, or make it available through a link on our websites. Good. Next slide, please. Now, the big challenge in neurodegeneration is, if you want to model disease and identify mechanisms, is that there's not a single coherent data set where you would have all the measurables in one experimental setting, or all the measurements in one setting. We don't have gene expression plus genetics plus microRNAs plus proteomics plus cognitive readouts, so clinical readouts, and biomarker readouts. These data sets don't exist, not even in model organisms, not even in the mouse or so. We don't have that. And the big challenge is to glue heterogeneous data basically together, and to bring them together in a way that they can be mined as a whole. And that is actually what we are after. And that is what we see in the next slide, if you would see it, yeah. That we follow an integrative modeling approach in the absence of coherent data, and that is a model-driven approach where we use on one side data, on the other side we capture knowledge, bring it together using various modeling languages, and we will learn about two approaches today, that Shweta on one side and Alpha on the other side are following. And then we make that amenable for inferencing, reasoning, and hypothesis generation. And of course, a model can be validated by using external new incoming data, new incoming knowledge. So with that, I hope I have set the scene, and I would like to hand over then to Alpha. Can we switch to?
Yeah, yes, but I cannot see the slides here again. Or maybe we can share our slides from our side. Shall we try sharing our screen?
That
Yep.
would be that would be a good good solution. Give that a shot.
Okay. Does it work?
Yep.
Yeah, okay, perfect. Yeah, so so I am Alpha Tom Kodamulli, currently doing my PhD with Martin Hoffman Apitu. So today I will be mostly explaining how I am doing the neurodegenerative disease modeling using Bell at Sky. So yeah, so so mainly today I will be explaining about the in-house built Bell models and how we are integrating the genetic variance information into the Bell models and the general concept about the reasoner to validate the Bell models. And as we all discussed now, yeah, all the scientific knowledge are either reported as publications or drawn as pathway cartoons or simply as omics data. But that doesn't mean that it can be directly readable by a computer. So there comes the role of the biological expression language, that is Bell, which we usually which we often use to model our diseases. So what is Bell? Bell is a descriptive language for modeling the life science knowledge into a computable form. So which captures the causal and correlative relationship as triples, and where each entity is referenced to the standard vocabularies and ontologies. So if you are going in the next slide, so here you can see textual information like phosphorylation of glycogen synthase kinase 3 beta at threonine 668 increases the degradation of amyloid precursor protein, which can be directly converted to to Bell using its function. So here you can see the red red words, yeah, mainly represents the Bell function. So here you can see the GSK3 beta, which is the glycogen synthase kinase 3 beta, which is reference to the HG with the HGNC and having the function P. So P here means just the protein. So when you have all the your context or the knowledge encoded in the Bell, then you can directly convert or compile into a network form. So there you get the mainly the computable Bell model. So here is the main workflow that we follow here at Sky. So it's mainly we extract the knowledge from the literature. So it can be from PubMed or from our text mining tool SkyView and so on, and also from the pathway cartoons and also from the databases like KEGG and so on. Then we convert the scientific knowledge into Bell and we get the computable model. So in the next slides, I will mainly explain the inbuilt Bell model. So we have mainly 3 Bell models. So one is the Alzheimer's disease Bell models, the next one is the neuroinflammation, and the third one is the human brain pharmacome Bell model. So the first one is the Alzheimer's disease Bell model. So this was our first Bell model, which is mainly dealing with the modeling of the neurons in the context of the Alzheimer's disease. For that, we mainly focused on the what all are the normal mechanisms, what is happening in a normal neuron and what is happening in the diseased neuron. So the normal mechanisms like neuronal survival, synaptic transmission, etc., and the pathological mechanisms like oxidative stress, mitochondrial dysfunction, etc. And we were like after to identify the main mechanisms that is perturbed in the early stages of the disease. And next slide shows mainly the statistics from where we collected the data, and here you can see a clear bias between the literature. So for the normal Bell model, we could get only about 64 publications, whereas in the case of the diseased Bell model, we could be able to get about more than or around 300 articles. So here you can see a clear-cut bias towards the more work around the diseased mechanisms as compared to the normal activities when you are considering the number of publications itself. And this is also reflecting in the amount of the edges and the nodes that we have in the normal model as well as in the disease model. And the main analysis that we have done on this normal and the disease model, mainly we have compared the two models, and we have identified, okay, what is switching between the normal state and also in the disease state. So here you can see we are explaining with a cartoon how the neurotrophin signaling pathway is switching between the normal and the disease state. So here you can see here you can see one BDNF branch and also NGFR and NGF branch in the neurotrophin signaling pathway. And all the green ones are showing the normal, pathway. So the BDNF is activated by UCHL1, and which is again binding with NTRK2, which is again activated and increases the neuronal development and neuronal homeostasis. But what is happening in the disease state in this branch? The amyloid beta is coming into play, and which is directly inhibiting the UCHL1, which is halting the entire neuron development and neuron homeostasis. And whereas in the other branch, which is controlled by NGFR and NGF, it is in the normally the NGF is binding to the NGFR and it is ubiquitinated, whereas then it binds to the IKB-KB, which is activating the NF-KB1, which is increasing the neuronal survival. And what is happening in the disease state? The insoluble amyloid beta is getting bind to the NGFR, which is competitively binding with NGFR, and so that the NGF won't bind to the NGFR, and the entire pathway is also getting stopped. So here we are explaining how, or we are currently trying to see how one pathway is perturbed from a normal state to the disease state. So in the next slides, I will explain how we integrated the genetic variation data. So currently in the Bell, we can just encode the SNPs data, like, okay, one SNP is associated with a gene, and we also tried to see how we can interpret the genetic variation data for the comorbidity analysis between two diseases. So, okay, this is a very complicated figure, but what is the important thing is that, okay, from our Bell models, we found that Alzheimer's disease is mainly having a comorbidity association with type 2 diabetes. So mainly the mTOR signaling is playing a role for controlling the autophagy. So here you can see. So in one branch from the type 2 diabetes, mainly the insulin signaling pathway is controlling the mTOR, which is mainly in the normal case increasing the mTOR. And from the Alzheimer's point of view, or in the normal case, the AMPK is getting phosphorylated and it is inhibiting the mTOR. So it is regulated. In one pathway it is increasing, and in the other one it is decreasing. So the mTOR is somehow regulated, which is again regulating the autophagy, which is helping to removal of the amyloid beta. But in the case of diseased condition, we have seen from the GWAS studies that there are many SNPs associated with insulin gene and also with the STK11, which hinders the phosphorylation, which decreases the phosphorylation of AMPK, and thereby increasing or hyperactivating the mTOR signaling, which is directly decreasing the autophagy. So what is happening? So it will not, there is no autophagy. That means there is no removal of amyloid beta. And also we have found the CLU, there are some SNPs associated with CLU form. And in the normal case, there is the CLU, which is actually secreting the soluble CLU, which is helping in the amyloid beta clearance. But in the case of a deceased case, some SNPs are associated with CLU, which is forming another form of isoform, which is again decreasing the removal of amyloid beta. So we can find a comorbid association between type 2 diabetes and AD. But when we are if we could include the functional element based on the location of the SNP, like the SNP can be in exonic part or intronic part or in the promoter region or in the enhancer region. Based on that, the SNPs can have different effects in the expression of the gene. For example, the promoter can downregulate the expression of a gene, or the enhancer can also do the same, but the silencer SNP can upregulate the gene expression. So we are working together with Selvenda to improve the current Bell version so that we can include these genetic locations also of the SNP, so that we can have more functional reasoning with the SNPs over the Bell networks. The next Bell model was what we were mainly focusing were based on the neuroinflammation. So it was based on basically for comparing the human and mouse. So it is basically to see how much the mouse can mimic the genetic responses of human. So we were building two models based on the neuroinflammatory responses specific to the cell types like microglia, neurons, and astrocytes. So, and we compare based on the Bell triples or Bell statements, how much Bell statements are similar to the human when it is, and also how much Bell statements from mouse is similar to the human Bell statement. So this is what, and this is a currently ongoing task, and I don't have the results right now. So the next Bell model that we have used is the human brain pharmacome Bell model. So which is, what is human brain pharmacome is, it is a database where we have already the drug information about all NTD drugs, but what we mainly focused for the HPP AD Bell model is that we took only the information from the HPP database, only the 400 AD drug information, and also the interactions to its targets. And the interactions can be mainly inhibition, binding, or increase the target activity. And we have expanded the mechanism around the top 100 targets, and also included the specific NDD pathways targeted by these drugs. And this is the overall statistics that we have, almost 4,550 publications, and the nodes is almost 1,000 nodes and more than 25,000 edges. And this is one of the analysis that we have done with our HPP Bell model. So we were trying to identify the potent drug for AD by the target similarity search. So what we have done, we searched for all NTD drugs which have more number of targets with the already approved drugs of AD. So there are only 5 approved drugs for AD. So we found that the cyclosporine, which is a rheumatoid arthritis drug, has more number of targets with common to all of the AD approved drugs. Then we tried to explain how the cyclosporine can be repurposed or can inhibit the main activators of AD. So Martin, do you want to comment on this?
Yeah, maybe.
Maybe, yeah.
Maybe I should.
Yeah.
I should briefly comment. cyclosporin has been used more frequently in the past, and it is for us one of the examples where we make use of the human brain pharmacome encoded in BEL. As Alfar said, we have a huge drug target network that represents basically everything that's bioactive in the human brain, and a selected portion of that was modeled in BEL. What you see here is basically a explanatory model for the putative beneficial effects of cyclosporin, which are, of course, in the inflammatory context, not surprisingly, really. But when we tried now to confirm a association or a putative protective effect of cyclosporin in epidemiological data, we found out that cyclosporin was frequently used in the past, but more recently, the clinical use of cyclosporin has gone significantly down. So we have to look into really old clinical data in order to establish the association between a putative neuroprotective effect of cyclosporin administration and that is something that's currently ongoing also in the context of our Taxonomy project.
Yep. Okay, so the another one thing that we tackle at the technical side is the challenge of doing the automatic plausibility checks for the BEL model. So I will briefly explain the concept. So the main motivation that when we are having like one BEL statements, we do not know about the full knowledge about the entity. So we do not know whether the GSK3 beta can actually do the protein modification of the tau. So if the GSK3 beta is a kinase, which is the case, then okay, of course it can do a phosphorylation. So these all are, so we are trying to see whether what we have already encoded in the BEL is complete and correct, or is there any, like if the so here you can see the relation like increases, so this arrow mark mainly means the increases, but here you can see the same thing, but if it is like decrease, then the entity which can do is the can be a phosphatase, or maybe there can be some intermediate pathway like, okay, protein X is increasing Y and then it is doing a phosphorylation, but the curator who is doing the BEL coding won't be may not have the knowledge that, okay, there are some intermediate pathways. So we are trying to see whether the BEL statements that we have already encoded is complete and correct. So we make use of the interpro domains for identifying whether a protein really have a domain to do some sort of relationships in the object that it is already encoded. So what this is the main workflow, like we have the interpro database and we have the BEL statements database, and we make basically the validation rules to see whether the subject can actually do the modifications that is already encoded in the object. So the interpro can be the first idea that we can implement for the plausibility checks, and another supporting rule can be also make use of the EC number, because the EC number classification also has the kinases, phosphatases, and so on. So based on that, based on the EC numbers, we can also make the rule. So this is also an ongoing task. And the third supporting rule can be we can make use of the GO terms. So the GO terms also have something like kinase activity, dephosphorylation, and so on. So we are thinking of using the GO terms also as a supporting element for doing the plausibility kind of checkings. And with this, I like to conclude my talk. So yeah, so basically I summarized the talk that we are using the BEL modeling approach to identify and integrate the mechanism to understand the etiology of the diseases, and some of the main applications that we have done so far is the generation of the hypothesis and identifying the early mechanisms that is perturbed in the disease condition, and species comparison based on the Bell models, and also using the drugs as the molecular probes to reason over the drug-target interactions, and lastly on the automatic plausibility checking for the Bell network. So with this, I like to hand over to Shweta.
Yeah, okay. Give me a minute. I have to just switch on to the, sorry, to the slides I opened earlier.
Yeah, there.
Sorry for that. Okay. Yeah. So, I'm Shweta Bhagewadi, and I will be talking about how we are implementing semantic web applications for building disease models, and we call it here NeuroRDF. This is the outline of my presentation where I describe why we are focusing on neurodegenerative diseases. However, Martin already gave a brief introduction to it. He described briefly why we want to focus on NDD diseases. Then some of the in silico models that are existing, what we would like to do using NeuroRDF, and a use case analysis showing that how good this approach is. Yes. Our understanding about brain is about 50 years behind than any other organ. And this is already quite motivating for us to work on brain-related diseases. Dementia has been affecting throughout the world, and it is expected that by 2050, it's going to be double, that the number of people affected by dementia are going to be mostly in middle-income countries, low and middle-income countries, and the numbers will double. So first of all, it's chronic and irreversible. There are lack of approved, FDA-approved drugs. We don't have reliable biomarkers. And also we have high death rate. There's also limited availability of samples, which I tried to capture in August. You can see that if we take cancer, this is from Geo and Array Express. These are the two major databases which store gene expression information. So if you see for cancer, you have a lot of data, but cancers can have different indications within the whole domain. So what I try to do is look into lung and breast cancer. These are the two most, most affected diseases in comparison to the topmost two diseases that are in neurodegeneration. And you can clearly see that the data out there, when you see NDD domain and cancer domain, it's quite limited in NDD. If we want to build an in silico disease model, since we cannot have a lot of data from brain, of course, so what we would like to do, what we would want in such an in silico model, first of all, we would like to have genetic information, then we would like to have a gene expression information in it, protein information, information that has been published, because that's a lot of data out there, drug-related information, imaging information, of course, which also adds value to current in silico models, and pathway-related information. So this in silico model that we want to build should support as a knowledge-derived decision support system. And the disease models should have the capability to recapitulate prime biological properties, because dementia, especially Alzheimer's disease, is quite complex. And we should have data interpretation over large knowledge space. The reason is we are going to integrate different data that are heterogeneous in nature, and we need to have good interpretation between the data themselves. There have been several previous approaches that try to integrate data in semantic web, One is Bio to RDF, where they try to integrate 35 different resources. Linking open drug data, try to link drug bank and clinical trial information. Chem to Bio to RDF is another such approach where they try to include chemoinformatics information to investigate polypharmacology. Open Facts was a huge project where they try to ask competency questions for drug discovery. If we go in detail, where they try to gain some mechanistic information, Shin et al. Tried to identify common genes that are differentially regulated by volatile organic compounds in a specific group of patients. Ku et al. Integrated 9 prior knowledge resources, and they devised a framework which could enable mechanistic linkage for drug repurposing in SLE. Alzpharm also is a similar approach, but this was a more focused towards Alzheimer's. And they tried to obtain drug information. They have drug information and manually inferred hypotheses, using which they try to filter publications specific to donepezil drug to see its effect on treatment of AD patients. So this is all what we know that's existing. What we would like to propose here is to attack this problem, that is to build an in silico disease model by merging expertise from different resources. So we have several heterogeneous data, as I previously showed, genetic, literature, protein, and so on. And we want to bring a formalization to all this data such that it is unified into one framework. This will enable us to obtain more precise and meaningful results. However, if we need quality, output, there has to be manual inspection of the data that we include in such model for preciseness and quality. I will show a use case scenario for the same, but for Alzheimer's disease alone. This is our overall workflow. Currently, we have tried to integrate databases, literature information, and experimental data. As I previously mentioned in the previous slide, that we need good quality data to reduce noise. So we do some data curation and pre-processing, which is integrated together in RDF models. We store them in our internal repository and do some data mining and analysis. I will come to the last point at the end when I show use case application, how we actually did the analysis. So let's focus first on how we collected the data and what curation and pre-processing steps we carried out. The first resource we included is from database, that's human brain protein-protein interactions. We assembled this from 21 databases that are being curated for human brain region specificity. This is, of course, manually curated such that we retain only true positives and we filter out all that is noise. This resource, we have about 7,000 genes and 45,000 protein-protein interactions that are occurring in 15 brain regions. Out of these 45,000, about 36,000 are found to occur in specific brain regions. The reason is that sometimes the authors mention that this is in the whole brain. So that's why there is the last sentence, specific brain regions. The second database resource is from Pathway. Currently, we are using Reactome. Reactome provides as a web service which we can automatically extract pathway information for each of the genes that we have in our model. So what we try to do is we have about 151 genes in our knowledge base. For each of these genes, we try to extract pathway information. And currently, we have about 280 unique pathways for 151 genes. Coming to the second resource, where we try to extract literature-derived information, Martin already gave a brief overview about our text mining expertise. We try to use ProMiner, run it over the whole midline. Using our tool Skyview API, we retrieve relations where they say, okay, this gene is upregulated in this disease, or gene A interacts with gene B. We try to extract such relations, we manually curate them to filter, to only retain true positives, and these true positives which build a network. Currently, we have protein-protein interaction network and microRNA target interaction networks that we have extracted from abstracts and full text. In case of microRNA target interactions, currently this is only from abstracts. Thus, we have only 29 miRNAs that is participating in 111 relations with 28 genes proteins. Whereas in case of PPI, since it is from full text, we have 303 genes and 339 interactions, along with information that how many of them are occurring in vitro, that's about 167, and there are 2 evidences that occur in vivo. Of course, they don't match the numbers 339 because not all the interactions provide in vivo or in vitro information. The third data resource is gene expression data for us. This is a very big workflow that we are working on. We downloaded all Geo and RxPress experiments into our internal storage called GenDB. This is our database. We run quality control analysis where we have machine learning algorithms to determine the quality of samples. And we apply metadata curation. Metadata curation here, I would like to talk a few sentences about it. Certain experiment has information such as from which patient it has been taken, what was the gender of the patient, what was the age of the patient, was it from hippocampus, and so on. All this information is available, but not directly in G or RxPress. Partially, yes, but not for all experiments. Most of the informations about experimental samples are provided in the associated publications. So we invested a lot of effort to capture all this information, going through the publications and attaching that to each and every experiment samples. This is already now in our GenDB. Once we have this, we can query any number of data sets for, let's say we want Alzheimer, give me all Alzheimer data sets that are that contain samples from hippocampus. So we can query. But for today's presentation, I will give only one. I will use only one data set, just as an example, to show how powerful NeuroRDF can be. So we try to use GSE 1297, run an R analysis on it, and try to extract differentially expressed gene using SAM. Here we consider top 1,000 genes that has a good p-value. And yeah, sorry. I think I talked already about that a lot. I will go further. Now we have all the data. And the important thing is we maintain a unified namespaces for each of these heterogeneous data resources. So we try to use common namespaces that can be used by academia anywhere. And we have published this in our website as well. So you can reuse the schemas, reuse the namespaces, and that should help to integrate, even if you want to integrate with our NeuroRDF data resource directly. Some of them, we could not find any namespaces, so we added a dummy namespace called Sky. And we also try to maintain provenance information from the data we collected, because it keeps growing, and we need to have the information, when was it downloaded, what was the information that was downloaded 2 years ago, and so on. So this we do it for each and every data resource. These are, this is the RDF schema for protein-protein interaction network, and we use the namespace that I just described in one of the previous slides. So you can see that the 2 proteins are connected by, if you see in the middle, that's core interaction, that's using the core ontology. We try to capture brain region information, what database it came from, and what is the evidence that supports this interaction. Similarly, we do it for pathway and also for literature derived. Here you can see that there is something called primary evidence, from which sentence this information has been extracted. From which sentence we know that there is protein A interacts with protein B. And this is one for the microRNA target interaction. It's similar to the literature, but doesn't have so much detailed information currently because we are not done with the full text yet. Sorry. And this is for microarray. Of course, it's quite huge because we try to capture all possible information so that we can do analysis, meta-analysis, the way we want to. So here we have a sample. We try to attach age, gender, from which organism it was taken, what was the platform, what was the expression value for that probe attached in this sample, and so on. Additionally, we try to attach SAM analysis results on the left-hand side, if you see. But of course, this can be changed with different statistical analysis that we want. So this is very flexible. We can change the analysis, and the only thing we have to do here is change the schema slightly. So now we have the data, we collected the data, we curated the data, pre-processed in the format we want, stored it into a schema that I just showed. Now we want to integrate them. How do we do it? The important thing is that the namespaces used between these data resources are the same, especially the gene. If you see that the gene resource on the left-hand side below, that's for the microarray, and on the right-hand side above, for the literature-derived informations are the same. We use the same namespaces. And once we have that, it's very easy to query in an integrative manner. These are the statistics of the resources we have currently in our triple store, and you can see the sizes. Okay. So we are done with data collection, curation, and storage in an integrative manner. Now we want to do some analysis on top of it. So here is an example what we wanted to do. We focused on Alzheimer's disease because amyloid beta plaques hypothesis has not been successful in late phase drug trials, and I think about 11 drugs failed recently. We need to understand, we need to have a better understanding of how the pathophysiological mechanism that leads to neurotoxicity in case of Alzheimer's. However, we didn't want to directly attack APP, that's the protein, but we wanted to see if there is some indirect mechanism that causes a beta accumulation. So how we did it? First, we wanted to ask some questions. We tried to talk to some of the disease experts and say, okay, what kind of questions would you like to ask to such a model? And they said, yeah, let's see what are the common genes between healthy and diseased. I think Alpha very nicely showed that there is little overlap between normal and diseased because it's more biased towards disease, so you have more information in diseased. And we would like to see, okay, if microRNAs participate in some of the post-transcription modulation, yeah. Are these genes up or down regulated in at least one of the gene expression studies? In this case, we used one. We chose the one that had that was very focused on Alzheimer's disease. And are there any local neighborhood cohesiveness? Do these genes or microRNAs participate in pathways that are linked to neurodegeneration? So these were the set of questions we fixed to, just to show an example. We implemented that in our triple store. We queried using Sparkle. The example on right-hand side shows a snippet of our Sparkle query that we use. This is for the to check how many genes are common between healthy and diseased. Once we have that, we built a very nice table showing, okay, there were about 13 genes that were intersecting between diseased and healthy. And when we filtered that to see how many microRNAs are targeting, we retain only 5 genes. Then we try to capture fold chain information and checked if their first neighbors in a network are differentially expressed. Which of these are really differentially expressed? And you can see the list on the right-hand side here, that these are the genes which are first neighbors of APP that are differentially expressed. However, this was not so intuitive. So what we try to do is we try to build a network around it and see, okay, not directly focusing on APP, but some other gene around APP. So we built such a network using the genes here and in the first column and in the last column of this table. We try to do some literature research around it. So if you see the numbers on the edges are the number of publications that support this interaction. And the numbers in brackets are the ones that support these interactions only in human.
Hello,
And
Shweta. I need to interrupt for just 1 second. We only have 4 minutes remaining and have a couple of questions we need to go to.
okay, I think I'm done. I need one more slide and I'm done.
Very good. Go.
Okay, so we try to focus on this triad and this triad here. There's only one human publication, and there is no human publication for this triad. And when we looked into those that were even supported by mouse, we saw that binding of tyrosine phosphorylated LRP1 to HSC1 transfers the HSC1 to plasma membrane for undergoing phosphorylation, and thereby activating RAS, and that's a possible cause of AD. And similarly, for the second triad, we found information that these somehow interact and cause neurotoxicity. Okay, I think I can stop with limitation and future work, since we have only 2 minutes. Or should I proceed, Alden?
Very good. We'll go. We have a couple of questions. The first one I'll read is, as you are drawing your data broadly from the literature, do you do any pre-filtering to address the source of data? For example, animal versus human data, in vivo versus in vitro, etc.
This is more a question of Bell model, or this is more a question about the NeuroRDF? Sorry for that.
I think I can answer that. We are doing extensive curation. That means we have a clear assignment of species specificity and the origin of the samples and the constraints that apply to the experimental setup, so that we can actually track back to organisms. We do also, as Alpha mentioned, species-specific comparisons in the neuroinflammatory field. We are modeling actually a mouse against human in the neuroinflammatory field, for the reason that there is an issue in neurology in general, to what extent the mice, and in genetically modified mice in particular, are good models or not for neurodegeneration. That's why we keep things really separate.
Very good. I don't think we have time for the last question, so I will conclude this by thanking our presenters. That was a great presentation, and we will not be holding another webinar in December this year. We are working on the schedule for next year, so please check back with us on that. Thanks again to everyone who joined us and everyone who presented. Have a peaceful remainder of 2014, and best of luck for 2015, everyone. Thanks very much. Signing off.
Thanks.
Bye.
Thanks. Bye-bye.